Again, I was asked to speak about precision microbiome in healthcare, or the role of the microbiome in precision healthcare is probably more effective. I'm a professor at University of California, San Diego, in the Department of Pediatrics and Oceanography, which is kind of odd, but we do run research programs in human health, clinical studies, the microbiome, and many other areas. And I do have disclosures that are associated with this program. So, understanding the clinical translation of the microbiome, I like to start this concept with the idea of three M's. This is the concept of mechanisms, metrics, and modifiers. This is a way of structuring our understanding of how the microbiome can be integrated into precision health or precision medicine. So, starting with mechanisms, we are predominantly trying to identify how microbes interact with the body and how they can be used to identify drug targets. So one example I illustrate here is the use of oral antibiotics, which we've identified can disrupt microbial nitrogen cycling in the oral cavity, leading to a change in nitrogen speciation in the bloodstream, which can alter blood pressure, causing hypertension. This mechanism allows us to create a direct link between microbial disruption in the oral cavity and hypertension, providing an opportunity for drug development. Metrics are associated with trying to identify microbial features, species, functional genes, metabolites, proteins, transcripts, etc., that are predictive of risk or response to a intervention or diagnostic of disease. And then we have modifiers. These are proven strategies to manipulate the microbiome. Think fecal microbiome transplants, phage therapy, etc., etc., which have been highly effective in allowing us to manipulate that microbial community and utilize it to make functional changes in human health. So, as I don't have long, I'm going to give a few key examples of how our research has contributed to the three M's. The first one is almost 10 years old now, or probably more than 10 years old, where we were able, working with Vanessa Leone at the University of Chicago, to identify structures associated with microbial presence that dictate host outcome. So this is a concept of mechanism. The role in which diet or influences, say, cardiometabolic diseases such as obesity, can be predicated on the presence of a microbial community. A germ-free animal up on the top here, whether it's consuming a low-fat diet or a high saturated fat, high sugar diet, it doesn't really matter. As long as it's consuming the same number of calories, you'll get a similar circadian functional dynamic inside the animal's organs. The brain will be in sync with the liver, and we will see the same kind of metabolic phenotype, the same amount of weight gain. But in the presence of a microbial community, i.e., what we refer to as conventionally raised organisms, the type of diet you feed the animal has very particular impacts upon the circadian function of that microbial community, i.e., its functional traits over time during the day, which will influence the types of metabolites that microbial community pumps out into the bloodstream at any given time. What we're able to identify here is that a high saturated fat, high sugar diet creates aberrant microbial changes over time, which lead to an increase about an hour after food consumption in the level of hydrogen sulfide in the bloodstream, which corresponds very well to an increase in hydrogen sulfide producing bacteria in the gut, which can lead to a disruption of the transcription of clock genes in the liver, host clock genes, that is, which leads to the liver perceiving time differently from the brain, which alters the adipose tissue production by changing hepatic hormone activity and also leading to an increase in appetite. That's a mechanism, right? Once we understand how the intervention influences the host through the microbiome, we can potentially intervene and create new drug targets. Another great illustration is work that we've done with Ben Shogan at the University of Chicago as well, back from the days when I was there, where we demonstrate that removing a colorectal tumor, for example, from the gut can lead to disruptions in the gut microbiome, potentially by the introduction of oxygen or by the pumping of antibiotics into the body. And this can lead to, now I've stitched the gut back up and you form what's known as an anastomotic junction. The body starts to now suck nutrients out of the gastrointestinal cavity. The only bugs that have survived the onslaught of oxygen and antibiotics will now change their phenotype in response to that nutrient starvation, forming a biofilm on the wound site, releasing an enzyme called collagenase, which breaks down the collagen matrix of the membrane and leads to what we refer to as an anastomotic leak. We've also demonstrated that these organisms, these collagenolithic bacteria, are increased by the presence of this standard American diet, or the SAD diet, SAD, which can lead to them being at low abundances in people consuming heavily large quantities of saturated fat, high sugar diets. And when we track these over the time schedule of surgery, pumping the body full of polyethylene glycol, for example, to wash out the gut, oral antibiotics, IV antibiotics, and then surgery, the relative abundance of these organisms increases dramatically, and they are the ones responsible for binding on that biofilm in the wound site. We've demonstrated that these collagenolithic bacteria and the presence of colorectal cancer cells post-colorectomy in an animal model will play a significant role in increasing the likelihood of the development of a perianastomotic tumor. What does this mean? This means that these collagenolithic bacteria are degrading the membrane at the wound site for the in the animal's gut, which leads to these colorectal cancer cells being able to transmit into the membrane of the animal, forming a tumor, which can lead to, in the case of a human, recurrence of the colorectal cancer. And we see that with a Western diet here down in the plus marking, we get almost 4 orders of magnitude increase in the abundance of, in this case, Enterococcus faecalis, which is collagenolithic, and the presence of antibiotics and surgery, E. faecalis, and a Western diet can lead to almost a 90% increased likelihood of recurrence of colorectal cancer in this animal model. We are currently running clinical studies which will identify whether a diet intervention can eradicate the likelihood that these collagenolithic organisms will cause recurrence. And we have very good data to support that hypothesis. One of my favorite studies, if I've got a few minutes left, is to just talk about our program METS, which is led by Lara Douglas from the University of South Africa in Pretoria, and Gertrude Eckler-Mansar, who's a project scientist working in our lab, where they are able to provide over a period of 15 years microbiome profiling over 2,500 people of African descent across 5 different countries. And like we were hearing before in some of the earlier presentations, the microbial diversity of these countries is very significantly different. And Ghana and South Africa, where we're dealing with rural communities who consume very high levels of fiber, have very much significantly greater Shannon diversity index, alpha diversity for the microbial community. We're also able to take microbial profiles and use them as metrics to predict outcomes in this population because we've been able to follow them for so long. So, for example, while we can predict which country you come from with very high accuracy, we're also able to predict your risk of diabetes and blood glucose with a quite high accuracy, at least 10 years prior to the onset of the disease. Obesity and hypertension less so. Interestingly, birth sex can be predicted, but only prior to menopause. Post-menopause, it's less easy for us to predict a birth sex woman versus a birth sex man, probably due to the influence of female hormone levels on the bacterial community during menstruation. We are currently running a nutrition precision health cohort, which we launched 3 years ago, and we're still recruiting populations through the All of Us program for the NIH. I'm a co-chair of this program, but also the lead for the University of California Microbiome Center. And if you're interested in this study, please take a screenshot or snap of this QR code, and it can lead you to information about our program. The program in itself is really designed to allow us to understand the impact of diet on human physiology and the potential role of the microbiome in mediating that outcome. Thank you very much for listening. I always like to say eat well and thank the people for funding our work. And if you are interested in any of this research, last year we were involved in developing a documentary called Hack Your Health, The Secrets of Your Gut on Netflix. It's still available, where you can find out some really interesting facts about the human microbiome and human health. And thank you for my team.